Um, Hi everyone, I'm Yeu-Chern Harn. Today the topics I'm going to talk about is deconvolving molecular signatures of interactions between microbial colonies. I would like to thank the conference organizer for providing this opportunity for me to talk about works. Finding metabolites involved in microbial colony interactions is important. Not only because we can know more about the metabolism network underneath the interactions, but also it is these metabolites are a potential source of novel drugs. However, few studies have investigated these interactions. And here, the metabolites we are interested in are of molecular weight less than or equal to 5K delta. In these three pictures, there are two strains of Streptomyces growing together. You can see when they interact with each other, the colony morphology changes as the underlying molecular change. Multi-imaging mass spectrometry is a technology that helps identify these metabolites, and I will abbreviate it as imaging INS. This is an illustration of the experiment of microbial interaction using INS. This is an agar plate where we grow two bacterial colonies. And in INS setting, this plate is divided into many rectangle grid cells. So we have a multi-mass spectrum in each cell. So the mass spectrum has the number of ions for each particular mass-to-charge ratio, which can be viewed as molecular weight. The spectrum can be viewed as molecular signals. So from all the spectrum in all grid cells, we can infer what molecules are present in any grid cells. So we can say IMS is a technology that helps find out the molecules and their spatial distribution. So a single molecule generates its own spectrum. For example, this is a molecule called surfactant, which is produced by some bacterial strains, and it's their molecular mass spectrum. However, in most cases, there are usually multiple molecules present in a grid cell. Therefore, multiple spectrum convolve in a grid cell. So we have a convolution problem. Can we learn the spectrum of individual molecules from the convolved spectrum in each cell? We present a method called molecular dictionary learning, abbreviated as MODEL. This method decomposes IMS data into two components. One is molecular signature spectrum, and another is abundance of these molecules. Usually, we do not know what molecules the bacteria are producing or are in a sample. Or even in cases, we might know what molecules are. We do not know what their spectra are. Therefore, we model a dictionary to be a collection of molecular spectra. This dictionary is what our method model aims to learn. So here is a dictionary referred as D, and where these molecular species M1, M2, M3 represent individual molecules, and the peaks shown in blue represent the individual spectrum for these molecular species. So a dictionary is a matrix of all the spectrum of all these individual molecules. A dictionary element is a column in this dictionary. An abundance vector weights each molecular species contribution. A spectrum in a grid cell is referred as a Y vector, and a Y vector is composed of the contributions from each molecular species. And we know a single molecule cannot generate all MOVZ values in IMS data. And in fact, we know from MODI mechanism that it prefers particular ion types, and they are shown in these two tables. So from these two tables, we can construct the pattern, a dictionary pattern Q on the right-hand side that has most entries zeros. So the dark point here means the non-zero entries, and all other entries are set to be zero. Importantly, this pattern only decides the sparsity of the dictionary. The non-zero entries are still treated as the parameters that need to be learned. So here is our model for a spectrum, a grid cell. This formulation relates a spectrum to a dictionary and an abundance. A spectrum Y can be seen as a weighted combination of different dictionary elements. So to learn abundance and dictionary elements means to decompose Y back into dictionary elements and their weights. In this model, we can observe the spectra in all grid cells are exchangeable and IID. That means there are independent and identically distributed random variables. So there's no special relationship, and this is a single grid cell model. We could set D to include many molecular spectra, but only a subset of them are measured in one grid cell. Similarly, a molecule could only generate a subset of ion types. To encode this kind of sparsity assumption, we put a Laplace prior, which has been extensively used to introduce sparsity on abundance and on dictionary, such that the molecules appearing in grid cells and ion types for molecule become sparse. So from all the spectra, since the IMS data is in all spectra across the grid cells, it is natural to assume IMS measurement at nearby cells have similar abundances. Therefore, we put the L2 square penalty on differences between neighbors. So this is the object function model aims to optimize. Model finds dictionary and abundance such that this function is maximized. We can see there's a balance between having our model more like data and more confined to the constraints. By putting different hyperparameters, lambda, phi, and theta, we can put different ratio of importance on the constraints. This is the outline of our algorithm. This algorithm ensures a good optimization property that is biconvex. So we can find the global optimum solution of abundance given dictionary and the global optimum solution of dictionary given abundances. Here I'm going to introduce the ways we measure our performance on dictionary recovery. For a recovered dictionary, if we know the ground truth dictionary, we can compare all entries in the dictionary if the dictionary size is small. Or if the size is large, we can use cosine similarity to compute agreements between pairs of dictionary elements. So in one of our synthetic example experiment, this experiment has multiple molecular species share the same M over Z value, and we want to show our method can deconvolve individual molecular species. We set the ground truth dictionary like this, and we set to simulate the characteristic that at nearby location have similar abundances. We set ground truth abundance vectors by first choosing a center grid cell, and we set the abundance for molecular. Then we diffuse its abundance across grid cells. Here is the comparison, entry by entry comparison of different methods. We can see our method can decompose all individual moleculars, while other methods such as NN and F, non-negative matrix factorization, and PLSA, probabilistic latent semantic analysis, cannot decompose well in this experiment. In another experiment, we set a larger scale experiment, set a large dictionary, and we set ground truth abundance vectors by the diffusion method, and then we randomly set some values to zeros, such that it simulates not all grid cells have all molecules. In terms of the number of molecular species, we set other methods directly to 20, our ground truth number. For our method, can learn by held-out validation automatically. This is the statistics of cosine similarity from different methods. We expect to see the diagonal entry to be high, such that the matched dictionary elements agree with each other, and the off-diagonal entries to be small, such that the mismatched elements disagree with each other. Table A comes from the comparison of the ground truth dictionary to itself. So it has the theoretical upper bound for the diagonal entries and the theoretical lower bound for the off-diagonal entries. Table B, C, and D comes from the comparison of the ground truth dictionary to the dictionary from sparse POSA, sparse NNF, and model. We can see among all, model has the largest value in diagonal entries and smallest value in off-diagonal entries. In the real data sample consists of two strains, bacterial strains, Bacillus cereus and Bacillus subtilis. Our method recovers the whole dictionary from the sample. In order to verify it, we show it can recover dictionary element of molecule surfactant. Surfactant, because previous studies on Bacillus subtilis show it produces surfactant, and surfactant is detectable in IMS. So we expect to see them present in our sample. We construct the dictionary element, the ground truth dictionary element of surfactant with experiment with purified surfactant. We then compare our dictionary element recovered from IMS data with the ground truth dictionary element. This is the comparison result in the negative mode and in the positive mode, which are both experimental setting in IMS. These results are for the surfactant C14 and surfactant C15. In both results, our method can recover the ground truth dictionary element. In fact, our method model shown in light blue bar are the closest one to the ground truth, shown in dark blue block. Also, our method does not have any false positive values. And compared to other methods, they have a lot of false positive values, and they cannot recover the ground truth dictionary as well as ours. So in conclusion, we present a method called model. It can deconvolve MOVZ values into molecular spectrum and their abundances. It models the characteristic of IMS data, such as it is a count-based data, it has spatial relationship, and it prefers particular ion type. And it can learn the number of molecules automatically. Compared to prior approaches, our method provides superior performance in terms of recovering molecular signatures. I would like to thank my advisors and other co-authors in this work, and I would like to thank ISCB providing my traveling funding. Thank everyone for listening.